This is the same slide I showed yesterday with one small addition. In chapter number 14, there's a fairly long list of study guide questions. We're not going to get to most of that material on this particular exam. So it's really the first 8 study guide questions that are relevant as far as the exam is concerned. And we will finish up likely today with the material for the exam, so that'll give us a little bit of break before Monday. So we'll see how we do today. Any questions? So one of the things that I really want you to be comfortable with is this idea of gene expression and what it means for a gene to be expressed. Those of you that had lab yesterday and were successful inserted a foreign gene for green fluorescent protein into E. coli cells. They previously didn't have that gene. And what's kind of remarkable about this is that those cells were able to take that gene and now express it. The reason you know that that gene is being expressed is they're able to do something they couldn't previously do, glow under UV light. So what does gene expression actually entail? Well, the central dogma really tells you what gene expression is all about. DNA has to first be transcribed into messenger RNA. So the information encoded into these A, T's, G's, and C's on a strand of DNA has to first be copied into a complementary RNA molecule. That particular RNA molecule is called messenger RNA. And that process is called transcription. And then that copy will have to then migrate out of the nucleus, if it's a eukaryotic cell, moving through the nuclear pores into the cytoplasm. And there it's going to hook up with ribosomes, and the genetic information encoded into the message is now going to be read or translated into a specific sequence of amino acids joined together by peptide bonds. Okay? There's one thing missing here in this particular abbreviated diagram showing you what gene expression is all about. And what it doesn't show you is how you get from here information to amino acids being hooked together. And that's something we'll talk about for the next exam. One of the things that's missing so far here is where do all of those individual amino acids come from? Your diet. But more importantly, how are they then hooked together to make a polypeptide chain? What's happening on that ribosome that allows this information to be turned into this tangible thing, this polypeptide chain, which folds up and does something important for the cell? In your particular case of your transformation, the polypeptide chain for green fluorescent protein now absorbs ultraviolet light. Okay? So, gene expression entails the processes of transcription of a gene and then translation to make a polypeptide chain. The concept of a gene has undergone some really kind of important evolution over a period of time. One of the earliest attempts to define what a gene was done by 2 researchers working with mold, Beadle and Tatum. And they did some interesting experiments that suggested that genes were information and that genes provided the information to make an enzyme. So the original kind of functional concept of a gene was one gene equals information to make one enzyme. That particular realization was modified a little bit later on because as people studied enzymes, they realized that sometimes enzymes are made up of more than one polypeptide chain. Hemoglobin isn't an enzyme, but it's a good example of this particular kind of phenomenon. Remember that hemoglobin consists of 4 polypeptide chains, and all of those interact with one another to form a functional hemoglobin molecule. Remember what we call that level of organization where you have multiple polypeptide chains interacting with one another to make a functional molecule? Not primary, is it? Not secondary. Not tertiary. It is quaternary structure. Okay? So people soon realized that if proteins had quaternary structure, that would require multiple genes to encode that particular information. In the case of hemoglobin, there are two genes. There's a gene for the alpha chain, and there is a gene for the beta chain of hemoglobin. Functional hemoglobin consists of two alpha chains and two beta chains all interacting together. So fairly soon, this concept of a gene was modified and said, whoops, one gene, one polypeptide chain. And that's kind of the functional understanding we're using at this point in time of what a gene represents. Later on, we're going to see that not all genes encode polypeptides. There are some genes that encode other RNA molecules besides messenger RNA. Messenger RNA encodes information for polypeptides. But there are other kinds of RNA besides messenger RNA. What are some other types of RNA? Transfer RNA and ribosomal RNA. Okay? And as it turns out, there are genes for all of the different transfer RNAs. There are genes for all of the different ribosomal RNAs, as well as genes for the various messenger RNAs that then encode information for polypeptide chains. So a later modification of the gene concept was one gene equals one cistron. A cistron is a functional RNA molecule. Now, most genes encode information for messenger RNAs and therefore encode information for polypeptide chains. But in your genome, there are genes for transfer RNAs, and there are genes for ribosomal RNAs. And as it turns out, there are some other types of RNAs you probably haven't heard before. We'll talk about those later on. But there are genes for those other types of RNA as well. So start thinking in terms of this relationship between genes encoding RNA sequences. Now, another thing we're going to talk about is that something that makes this concept of a gene even more complicated is that genes in some cells in your body can also be rearranged and reorganized. This particular phenomenon is called gene shuffling. And one of the phenomena that's associated with this gene shuffling is an idea called alternative RNA splicing. And so this is something we're going to talk about when we get into the next exam material as well. So bottom line is that this concept of a gene is fairly complicated. It's much more complicated than just information coding for an enzyme. And we're going to dig into some of this complication of what a gene is later on as we move through more information about gene expression and what that entails. But for right now, if you want to be thinking of one gene, one polypeptide, that's good working information to get us where we need to be so far, at least. Okay. So, if DNA contains genetic information, and DNA is made of nucleotides, and the genetic information encodes primarily information to make polypeptide chains, there's a coding system working here. Okay? So somehow letters, A, T's, G's, and C's within your chromosomes provide information that says amino acid, different amino acid, different amino acid. So there's a correlation somehow between A, T's, G's, and C's, encoded information, and product amino acids to make polypeptide chains. So what's the nature of this genetic code? How is the information stored within the A, T's, G's, and C's of your chromosomes? Well, there are several characteristics of the genetic code that we need to talk about and make sure you're comfortable with and understand. And one of the most basic ideas about the nature of the genetic code is that the genetic code is a triplet code. What does that mean? In a triplet code, 3 consecutive nucleotides, like an A, a T, and a G. 3 nucleotides right next to one another specifies the information for a particular amino acid. So there's this equivalency between 3 bases in DNA and a particular amino acid. We call this group of 3 consecutive nucleotides a codon. Well, there are 4 nucleotides, right? A, T's, G's, and C's. And if the genetic code is 3 nucleotides in size, then how many different combinations are there of 4 things taken 3 at a time? There are 4 cubed combinations. And so there are 64 distinct codons in the genetic code. Okay? But remember that there's this equivalency between codons and amino acids. How many amino acids are there? How many common amino acids are there? There are about 20 common amino acids. So immediately we see that there are more code words than there are distinct amino acids. Now, we can explain some of this difference between 64 code words and 20 amino acids by the fact that the genetic code has some punctuation marks built into it. And one of the punctuation marks that is built into the genetic code are codons that say stop. This is where the genetic information ends. Okay? Well, if there are stop punctuation marks, what else does there probably have to be? Start. Interestingly, the start punctuation mark in the genetic code also specifies a particular amino acid. So that's kind of interesting. The genetic code word that says start also encodes the genetic information for the amino acid methionine. So what does that imply? That implies that all genes encode information that says the first amino acid in that polypeptide chain is always going to be methionine. Interesting. All polypeptide chains begin with methionine. Now, as it turns out, once a polypeptide chain is produced, sometimes that initial amino acid or several others is cleaved off. And so the functional protein that actually does something in the cell may eventually not have that methionine amino acid. But when it's first produced, that first amino acid in the polypeptide chain is always this methionine amino acid, because the start code word says methionine goes here. Now, there are 3 stop codons, and they do not specify a particular amino acid. All those stop codons say is, this is where the information ends. So, now we've got 61 code words, 64 minus 3, for 20 amino acids. So what does that tell you? Well, that tells you that there is some redundancy built into the coding system. If we've got more code words than there are amino acids, then the implication is that some amino acids are encoded by more than one different codon. So there can be multiple codons that say amino acid phenylalanine. There can be multiple codons that say amino acid tyrosine. Now, when we look at the genetic code, one of the things we're going to see is there's a pattern in the variability of redundancy within the genetic code. That probably doesn't make any sense to you at all right now, but it will when I show it to you. The third aspect of the genetic code is that the genetic code is considered to be universal. What does that mean? That means that if we have a particular code word that specifies a particular amino acid in humans, that same code word specifies the same amino acid in a plant, or in a fungus, or in a bacteria. That all organisms use the same basic genetic code to store genetic information to make polypeptide chains. What does that imply about life? One of the implications of that is that all life had a common ancestry. And that once a genetic code was developed, it was retained or conserved in all of the resulting organisms that were offshoots of that basic initial ancestral form of life. Now, there are some practical applications to the fact that the genetic code is universal. If bacteria used a different genetic encoding system than jellyfish, would you be successful in transforming your bacteria in lab with the green fluorescent protein? No. Because you would give them a bunch of DNA sequences that in a jellyfish said, this is how you make a green fluorescent protein. But if a different genetic code was used in bacteria, they would look at that and it wouldn't make any sense at all. So the fact that we can transform cells and take DNA from one organism and put it in another organism and then produce a functional protein is really supportive evidence of the fact that the genetic code is universal. Another characteristic of the genetic code is that the genetic code is non-overlapping. What does that mean? Well, if I've got a sequence of DNA, this 6 nucleotide sequence would represent the information for 2 code words. Here's codon number 1, and here's codon number 2. And then this would represent the information for a particular amino acid, and this would represent the information for another amino acid. Okay? This is way it's happening if the code is non-overlapping. The nucleotides are read in distinct groups of 3. If we had an overlapping code, things would look different. This would be one code word. This would be a second code word. This would be a third code word, etc. Okay? So the genomes of organisms are set up so that the code words are read in distinct groups of 3. The nucleotides don't overlap when you generate genetic information for amino acids. So a triplet code, a universal code, a redundant code, and a non-overlapping code are kind of the characteristics of genetic coding information. Well, we said that in biology, in life, there are typically exceptions to rules. And there are some exceptions to these particular rules. We have not found an exception to a triplet code. We have not found any instances where there is a genetic coding system that involves one nucleotide specifying an amino acid, or 2 or 4. This seems to be a rule that isn't violated. Watson and Crick, when they discovered the structure of DNA, didn't ever say it in the paper, but they intuited that the genetic coding mechanism of their DNA molecule was a triplet code. How could they know that? Well, think about it logically for a second. If the genetic code were a singlet code, If one nucleotide specified one amino acid, how many amino acids could there be? Only 4. And there are 20 amino acids. So a singlet code wouldn't be nearly big enough to encode the genetic information that was needed. What about a doublet code? What if there were 2 nucleotides? How many combinations? 4 things taken 2 at a time. 4 squared. 16. Still not enough, right? 16 possible codons, 20 amino acids. Not enough. Triplet code, 64 possible combinations. More than we need. Quadruplet code, 4 to the 4th power, 256 combinations. Way, way more than we need. And so they had a pretty good understanding, even at the time, that it made sense that the genetic code was a triplet code. And we haven't found any exceptions to that rule. The genetic code is nearly universal. As it turns out, there are some minor situations where there are slight variations in the coding system in certain organisms. There are some ciliates that have minor changes in the standard genetic code compared to other organisms. There are some genes within mitochondrial and chloroplasts that have minor alterations in their genetic code that make them a little bit different than the normal genetic code we're used to seeing. But these are minor exceptions. By definition, if the genetic code is a triplet code, then it's got to be redundant. So we haven't found any exceptions to that. We have found some exceptions to the code being non-overlapping. We haven't found these in organisms, but we have found them in virus. Now, we haven't talked about virus yet, but technically viruses are not organisms. We'll talk about that distinction later on. One of the things that's important for you to understand about viruses is that they're really small. And that sometimes they have problems storing enough genetic information to encode the virus because they're so small. There are certain viruses that have been discovered that actually have an overlapping genetic code. Think about the implications of that for a second. If we've got 6 nucleotides and the genetic code is non-overlapping, how many amino acids can that encode? Just 2, right? But if the code is overlapping, we can have this be the information for amino acid, this. So we can squeeze more genetic information into the same set of 6 nucleotides if the code is overlapping. And so sometimes, in rare instances, there are certain viruses that we'll see that have an overlapping genetic code. Okay, here's the genetic code. Looks a little bit complicated. It's really not that bad once you first kind of dig through it a little bit. This little matrix right here has the 4 bases that can occupy the first position of the code word, the second position of the code word, and the third position of the code word. One of the things that you'll immediately see is that this particular genetic code is based upon the code words in the messenger RNA molecule. This does not represent the genetic code of the nucleotides in DNA. How do you know that this is RNA and not DNA? You have uracil in there, okay, instead of thymine. Okay. So in this genetic coding system, what we've decided that we're going to do in terms of defining the genetic code is not look at the code words in DNA, but look at the code words in the message that is made from the DNA. So this represents actually a codon. This is another one. This is another one. This is another one. So remember that code words represent RNAs and not DNAs. So here you can see that there is this start sequence, AUG, that basically says information begins here. And that start sequence also specifies a particular amino acid, methionine. So it does double duty, start and particular amino acid. Here are our 3 stop codons. So anytime a message contains one of these unique sequences, when that ribosome is translating that message into a protein, if it hits one of these stop sequences, it's done. Okay, so that's our stop punctuation mark. And then you can see that there are different amino acids specified by different codons in this genetic code. So phenylalanine, leucine, serine, tyrosine, cysteine, arginine, glutamic acid, aspartic acid. Okay? So this basically tells you what the equivalency is. In messenger RNA, GAU means aspartic acid goes here. AAC, a different amino acid. AGU, a different amino acid. Okay. When you were little and you were playing with friends, did you ever make a simple substitution code so you could write secret messages to one another? And you said, in our code, letter A is going to be Z, and letter B is going to be W. And if you had the cipher, if you had what the substitution was, you could write messages to one another and translate them. Well, that's basically what this is. This is information that was encoded into DNA in the form of A, G's, T's, and C's, copied into messenger RNAs, and then the ribosomes basically are going to read those code words in the message, and that will tell them which amino acid is hooked to which amino acid. These brackets right here represent points of redundancy. So what you'll see right here is that the amino acid glycine is specified by 4 different distinct codons. Arginine is also specified by 4 distinct codons. Histidine by 2. Okay? What pattern do you see in the redundancy?
Nucleotides are always the same.
First 2 are the same, so base 1, base 2. The variability is in the 3rd nucleotide of the code word in these redundancies. As it turns out, that's going to have some significance later on that we will come back and talk about. Normally, we would expect that an alteration in the coding system, if we turned one of these A or C's into a G, that would represent a mutation. And we would normally expect then that changing that C would change one of the code words, and therefore one of the amino acids in the polypeptide chain. But would it necessarily? Redundancy tells us that changing this 3rd nucleotide in DNA, which changes the 3rd code word in a codon, might not make any difference at all in the amino acid that's specified because of this redundancy of the code. So one of the things that redundancy does is it helps to protect the cell from adverse effects of random mutation. Because if the mutation occurs in the 3rd nucleotide of a code word, it might not make any difference at all. It might still specify the same amino acid. There are other significances about the redundancy of the code and that 3rd nucleotide that we'll pick up later on. So here's an interesting plant. This plant has been genetically modified. This is a tobacco plant. And what this particular tobacco plant can do is it can glow in the dark. Now, tobacco plants normally can't do that. This particular tobacco plant has been genetically modified using techniques not dissimilar to what you're doing in lab to contain a new novel gene. This isn't a green fluorescent protein gene because we're not shining ultraviolet radiation on it. If you drive by fields of these genetically modified tobacco plants at night, you'll just see them glowing. Why? because they've been genetically modified to have a firefly gene in them. And that particular firefly gene encodes a particular protein that allows them to do the same thing that fireflies do at night. So why are they doing that? Well, what you know is that particular gene is being expressed because the phenotype of this particular plant has been altered. This exactly illustrates what Dr. Morton mentioned to you the other day. Why do we insert green fluorescent protein genes into other organisms and make them glow green? Not just for the novelty of it, because if we couple another gene with the green fluorescent protein gene or the firefly gene, then that can give us a mechanism to tell us whether we were successful in transforming that organism. Because if this firefly gene is being expressed, then very likely the other genes that were transferred along with it are being expressed as well. So this firefly gene is being used as what we recall a reporter gene. And that's a very useful tool that we can use in genetic transformation experiments. So, the gene's being expressed, then that gene is being transcribed into a message, and the message is being translated into a protein. And this protein that's being produced has some function in the cell. And if this is a novel gene that the organism has been exposed to, that can then change the phenotype of the cell. So your E. coli are now over there expressing green fluorescent protein genes and making green fluorescent proteins. This idea of gene expression is really important if you understand the concept of totipotency. Fancy word, what does it mean? Basically what it means is that all of the cells in your body, except your gametes, your sperm and eggs, have the entire complement of chromosomes to make a whole organism. Every body cell or somatic cell that makes up your skin, your liver, your eyes, has the complete set of 46 chromosomes, 23 from mom and 23 from dad. And all of those particular bits of information define you as an individual and determine what your characteristics are. At fertilization, the 23 chromosomes from the sperm are united with the 23 chromosomes from the eggs to give you a 46 chromosome zygote, which has 23 pairs of chromosomes from each parent. This is diploid. These gametes are haploid. Remember those terms? So we all start out as a single cell. You're no longer a single cell. You're a multicellular organism consisting of well over 1 trillion cells. How do you get from 1 to 1 trillion? Your cells have to divide, right? And when the cells divide, the nucleus has to divide too. And that process of nuclear division is mitosis. And if mitosis is accompanied by cytoplasmic division, then over a period of time, the number of cells doubles every generation. One forms 2, 2 forms 4, 4, 8, etc. And eventually you get up to this trillion plus celled organism. But not all those cells are alike in an adult organism. So organisms not only grow, but they differentiate. Cells become specialized. And at some point in time, cells start diverging off from one another and go on different pathways to become muscle cells and liver cells and skin cells and brain cells. But they all have the same genetic information. They're all totipotent. So why do some cells become different than one another if they have the same instructions? Because during this process of differentiation, there also is differential gene expression. And at key times, some cells in your body have certain genes turned off, and other genes are turned on. And that orchestra of events is critical to happen in the right way at the right time if a functioning adult organism is going to develop out of that one single cell. So the regulation of gene expression is something that is really important and is really complicated. And that's something we will dig into after we first learn the basic mechanisms of transcription and translation, gene expression. Then we'll put a twist on that and talk about how can you turn on and turn off genes at certain key times in an organism's life cycle and in their developmental process. And that gets pretty hairy. We'll just scratch the surface on that. So, differential gene expression is involved both in embryonic development. At some point in time, you start forming limbs, hearts, and is accompanied also by specialization of tissues as those new body parts develop. One of the things that we've learned about gene expression is that hormones can have a very important effect on regulating gene expression. Good illustration with you and your bodies during development. At some point in time, males and females start producing different hormones. Testosterone, estrogen, progesterone. And as these cells are exposed to these particular hormone molecules, those molecules can then function to turn on and turn off genes and stimulate different developmental pathways for males and females. One of the ways that we can determine whether those pathways have been activated is that males and females start developing different secondary sexual characteristics. That is the result of the action of hormones on genes, many of which have been lying dormant your whole life up until that particular time. And then under the influence of the hormones, all of a sudden the cells start expressing new proteins, which alters phenotype and results in the difference in two sexes. Insects oftentimes go through a very interesting life cycle where their body form changes during the developmental process of the insect. We call that particular phenomenon metamorphosis. An egg first grows into a larvae, like a little caterpillar. And then at some point in time, the body of that caterpillar undergoes a dramatic change where it forms an adult moth or butterfly, for example. What's going on there? The egg cell, the larvae, and the moth butterfly all have the same genes. They all have that 23 from mom and 23 from dad if you're humans, or whatever the insect number is. They all have the same kinds and numbers of chromosomes. But gene expression starts changing, and first we're expressing larval proteins in the larval stages, and then later we switch over and shut down those larval proteins and start producing adult proteins. What causes that? Hormones that are produced are very, very important in regulating gene expression during metamorphosis. There are hormones called juvenile hormone, for example, that regulate gene expression. So it gets pretty complicated. Like I say, we'll scratch the surface and give you some insight into some of this. So we're just starting. Okay, so the first step in gene expression is taking the information in DNA and copying it into a messenger RNA molecule. That particular phenomenon is called transcription. If you're a eukaryotic cell, where does transcription take place? It takes place within the nucleus. What do we need for transcription to take place? Well, we need something to copy. So we need some DNA. We need a template. We also need some RNA nucleotides because we need to join those nucleotides together to form an RNA molecule. And we also need an enzyme that's actually going to hook those nucleotides together and bond them together. The enzyme that makes RNA during the process of transcription is called RNA polymerase. One of the things we're going to see that, especially in eukaryotic cells, typically this RNA polymerase enzyme is going to need some help in order to do this process of transcription. So there are a number of transcription factors that are required. Those transcription factors are protein molecules. So these are the basic requirements that you need for the process of transcription to occur. Remember that DNA is a double helix, and there are two strands. When we transcribe a gene, only one of those two strands is actually copied into a message. The strand that is copied is called the template strand. So, if I've got DNA, remember I said that sometimes the two strands of DNA are represented as the Watson strand and the Crick strand? So if this particular stretch of DNA had the genetic information for 3 genes, are they always on the same strand? Is this always the template strand? No. For one gene, the template strand might be the Watson strand. For another gene, the template strand might be the Crick strand. For another gene, it might be the Watson strand. So it's not like all the genetic information is encoded into just one half of the DNA molecule. Sometimes it's on one strand, sometimes it's on the other. But not both, okay? Not some of the information here and some of the information here. Okay, so here is what transcription looks like. Here is our DNA molecule. Here's our DNA molecule, 5' to 3' strand, 3' to 5' strand. Here is the basic information in here that will encode a messenger RNA. This particular region right here in light green is also a series of A, T's, G's, and C's, but it doesn't encode information for a message. This is kind of a docking site, if you like. This is a recognition site. And this is where the enzyme RNA polymerase positions itself on the DNA and basically lines up so it can then begin transcribing this particular information. That particular region is called a promoter. So promoter is a region of nucleotides in DNA that is recognized by the enzyme RNA polymerase. Once the RNA polymerase recognizes the promoter, the enzyme starts to pry apart these two strands from one another. So we're breaking hydrogen bonds. And in this particular case, it's this strand right here that is the template strand. So this is the strand that's going to be copied. So what happens? RNA polymerase now starts reading these nucleotides and assembling an RNA molecule that is complementary to this template strand. And like DNA polymerase, RNA polymerase is working in its 5' to 3' direction. And the messenger RNA that's being formed from the DNA is anti-parallel. So if this strand is 3' to 5', this is going to be the 5' end of the RNA, and this is going to be the 3' end down here. That can sometimes make things a little bit confusing. So, positioning of the RNA polymerase at the promoter. We begin transcription, and a RNA molecule is now being formed as the polymerase moves down this particular stretch of DNA, copying this template strand. And again, notice the anti-parallel nature of this template strand and the RNA molecule that's being formed. Once a termination or a stop codon is reached, then transcription is complete. The RNA molecule that's formed is released from the DNA. The polymerase molecule is released. And now this copy of RNA that's been produced can move out of the nucleus and into the cytoplasm and be translated into a polypeptide chain. This shows you the complementary base pairing that occurs as this strand right here is being read and converted into a complementary RNA molecule. So again, this is a 3' to 5' strand. The new RNA is being made in the 5' to 3' direction. And the reason that this particular strand can be assembled is that the base pairing rules apply to transcription as well as replication. So anytime there is a T here, the RNA nucleotide A will be placed across from it. Anytime there is an A in the DNA, uracil is going to be placed across from it. Those two nucleotides will be joined together by a phosphodiester bond, just like in DNA, and that's catalyzed by this RNA polymerase molecule. So there's a complementarity between the RNA and the DNA, but remember, uracil replaces thymine in RNA. Transcription cooks along pretty fast. We can make RNA molecules by transcription at a rate of about 30 to 60 nucleotides per second. So again, just think about the implications of that. 30 of these are strung together in 1,001. That's fast. As it turns out, there are many different types of RNA polymerase. There's a ton of different types of RNA polymerase in eukaryotes. In prokaryotes, there are 3 basic types of RNA polymerase. Polymerase 1 transcribes ribosomal RNA genes. Polymerase type 2 transcribes messenger RNA. And polymerase 3 transcribes transfer RNA genes. So if this were genetic information for a polypeptide chain, and this were a messenger RNA that were being formed, then that particular polymerase would be this type 2 polymerase. The promoters that are needed to position the RNA polymerase properly are made of about 100 nucleotides. And promoters can be found in various levels of strength. Some promoters are very strong, and some promoters are really pretty weak. So if we've got a strong promoter, that's really going to grab RNA polymerase very readily. And the stronger the promoter is, the more likely it is that that gene will be transcribed. If the promoter is kind of weak, then levels of transcription tend to be less than if the promoter is strong. In eukaryotes, some additional proteins are needed to help the RNA polymerase to bind to the promoter. And there's some interesting organization happening in the promoter of eukaryotic genes. So if this is a eukaryotic gene, this region is the promoter. That's where the RNA polymerase is going to attach. Eukaryotic promoters tend to have a sequence of nucleotides associated with them that are very rich in A's and T's. And these regions are called the Tata box. Out of time? Oh, sorry about that. Thanks for keeping me honest. Go back up here Thursday.